I'm from the computational biology program at Stone Kettering, and I'm very happy to be here today to present our recent work on the context-specific regulation by miR-155 and how it's possibly related to the alternative polynodulation mechanisms. First, as background, miR-155 is a small single-stranded RNA molecules which repress the expression levels of its target transcripts by binding to particular target sites within the 3' UTRs. And then those target sites usually involve complementary matches to the 2 to 8 seed regions of the miR-155 sequences, and that's how we usually define the target sites of a particular miR-155. So when we are talking about context-specific microregulation, we are basically asking this question, like I'm trying to show here with this really simple illustration, that is it possible that the same microarray could be regulating its target genes differently across different cell types, just like what we have seen in the transcription factors? And if that's true, if that's true, then what are the possible mechanisms? So recently, there have been some studies on these questions, although the results have been kind of conflicting with each other. For example, there's one paper from David Bartel's lab recently in which they are transfecting microarrays into several different cell lines and then measure the changes of expression levels of target genes as a metric for the extended regulation. So if you look at the results here, then you can see that all these extended regulations is actually quite consistent between each pair of cell types, and only very few of the target genes can be considered context-specific, like you're seeing those few dots in the darker colors. But on the other hand, one can still argue that because those microarrays are not natively expressed in those cell types, and also because when you transfect them, the expression levels of those microarrays are like hundreds times higher than the native physiological levels. So maybe this setup is not very relevant to the actual physiological conditions. And if we switch to something more biological relevant, then maybe we can actually observe a different results. And that's why we turn to look at the miR-155 microarray, which is already known to be a very important microarray in the immune systems. And actually, as you can see here, for many different types of immune cells, once you activate them, then you will see a dramatic increase in the expression levels of miR-155, whereas for the other microarrays, there's barely any change. And also, if you knock out miR-155 from mice and then expose them to the bacterial infections, then you see that they all die within a very short time, indicating their massive defects introduced by the lack of miR-155 in their immune systems. So because this microarray has important functions in so many different types of cells with distinct biological roles in the immune system, I think it's fair to suggest that it might also be actually regulating different sets of target genes, which could provide the perfect system for a question. And I'd like to talk a little more about our hypothesis about what's the mechanisms for the context-specific microregulation. Here we then focus on one mechanism, alternative polyendonylation, which is basically saying that in the same 3' UTR, there could be multiple cleavage sites at the to form the end and poly A tails, and the usage of those sites is actually quite diverse across different types of tissues. So basically that means in some kind of styles, you'll be getting shorter isoforms, whereas in the other cell types, you'll be getting longer isoforms for the same 3' UTR. And actually, we already know that upon T-cell activation, there's actually a very significant shift of many 3' UTRs towards the shorter isoforms, which is actually happening at the same time as the increase in the 155 expression levels. So how is possibly related between the two mechanisms then? Well, I mean, if you look at this example, then I guess it's very easy to suggest that, for example, for the short isoform, which don't have any target sites for the microarray, and the longer isoform, which has a target sites in this distal region, and then if like one cell type only express this isoform as a major isoform, and the other isoform is only like use the other cell type, then it should be very clear that this gene will only be regulated in, only regulated in one cell type instead of the other because the target site just no longer there. And then we call them the differential regulation of the distal sites. And then on the other hand, if you look at another comparison, like the third and fourth isoforms I'm showing here, then Both isoforms share the same target sites, but because of different lengths, then the relative positions of those target sites within 3' TR could also be different. And there's actually already established that the relative positions within 3' TR can actually really change the regulatory efficiencies of the same micro target sites. And we call them the differential regulation for the proximal target sites. So those are the two possible hypotheses I'm talking about. And of course, it's also possible that the same microarrays, the same 3' TR isoforms could be differentially regulated as well, independent of APA mechanism. And here we want to focus on one thing about possible interactions with the RNA binding proteins, because it's already known in some studies that some RNA binding proteins could be opening up the secondary structures of mRNA transcripts, and then therefore exposing the target sites for the microarrays. And sometimes it's the other way around, they're actually blocking the target sites within 3' TR, therefore inhibiting the microregulation. And also because expression patterns of those microarrays are highly diverse across different tissues, so they could also contribute to the context specificity. And now we are going to talk about how we actually approach this question in the immune systems. We're using this technique called poly-A sequencing, which is basically we are using a primer sequence containing oligo-Ts to anchor to the poly-A tail. Therefore, all the sequencing reads we get from this primer will be mapped to exactly the end of the expressed 3' TRs. And then here's like what the actual data sets look like. We did the same experiments in 4 different types of immune cells. And then to relate them to the 155 regulation, we also did experiments both in the wild type and miR-155 knocked-out mice. And of course, this is in collaboration with the Rudensky lab at Stone Kettering. And if you look at this example here, which is for the 3' TR of this gene, Wink1, and it's actually a target gene 425, here's the target sites, you can actually see a lot of things going on here. For example, the simple thing is like in the annotation databases, there are only one 3' TR for this gene. But then if you look at our results here, then clearly there are these 2 more major 3' TR isoforms indicating by those 2 peaks in all those cell types. And then if you look at the abundances, the recounts for those isoforms, then you can see the usage of all those isoforms also quite different from each other. Sometimes the shorter ones are the major isoforms, sometimes the longer ones are the major isoforms in different cell types. And then when you compare between the wild type and 155 knockout samples, then you can see that sometimes this isoform containing the target sites will be upregulated after you knocked out 155, suggesting there's regulation for this isoform. But then that's not always the case. If you look at the same isoform, then in other cell types, it's just not changing at all between 2 conditions. And the other isoforms, despite the fact they all contain the same target size, they also could not be regulated. So we already see like some signals for our hypothesis, but then of course we want to describe those effects more quantitatively. And here we're using the generalized linear models to describe the differences in expressions and regulations. So the simplest case in the same cell type, we can simply compare between the knockout and wild type conditions. And then the differences in expression should show us the extent of miR-155 regulation. And then if we want to actually compare between 2 cell types, then we just simply add in another cell type factor. And then in this case, the interaction term in the generalized linear model will tell us about the differential regulation between 2 cell types. And of course, besides like doing this on the isoform level, if you just simply sum up all the reads from the multiple isoforms of the same UTR, then we can also do the same analysis, but this time on the gene level. So we actually start on the gene level analysis. So here you can see we define target genes. They have to have a target size, and they have to have the correct directional regulation, and then have a certain level of significance. If you look at the specific target size in each cell type, you can see they're always shared. They always share target genes between all the cells, but then in each cell type, you're also seeing those target genes specific to it. And if you look more closely, here we're plotting against the changes of expression, or in other words, extent of regulation in different cell types. And then in those scatter plots between each pair of cells, then you can see that although there are always genes along diagonal which are similarly regulated, we already see a fairly large fraction of genes which actually differentially regulated between each cell type. And you can, if you remember the early results I showed from other papers, and there's a clear, much larger fraction than what they said, which is saying maybe like the context-specific regulation is more prevalent in the like more biological system. And then if you look at actual target genes, that's specific, what they are, and then the goal analysis actually reveal that they're actually quite relevant to the biological functions of those cell types as well. For example, in dendritic cells, those specific genes actually involved in cytokine secretion. In macrophages, they're involved in chemotaxis. In B cell, they're involved in cytidine deamination, which is related to the antibody production, and T cell, they're related to differentiation and activation. All those suggest that all the context specificity we observed could also be related to the biological functions of the same cell types. And we also did same analysis on the isoform level. Here you can see the situation is actually really similar, that even the same single isoform could also be differentially regulated between cell types, suggesting that there could be the independent, the mechanism independent of APA that we suggested earlier. Sorry. But then you can still argue that to actually make those differences functional on the lysoform level, still in the end, they have to be somehow reflected on the gene level expressions. And we want to show some like more exploratory analysis on like how is the isoform changes relate to the gene level changes. Oh yeah, and here of course our collaborators have been picking up some isoforms which are interesting. Some of them are only regulated in B cells, and some of them are only regulated in dendritic cells. And you can see from their luciferase assays in both cell types, with one exception, everything else is highly consistent with the poly-A sequencing results, suggesting like, suggesting that our poly-A sequencing results are actually quite quantitative. And then, yeah, so for, as I just said, to explore how the isoform level differences are reflected on the gene level, we are going to go back to the two possible APA-dependent mechanisms I mentioned earlier. One is, if you still remember, the differential regulation of the proximal, of the distal target sites. Here, this on the left side, there's the gene level differential regulation, which you already seen before, the same thing. But then on the left side, I'm actually showing the differences in the 3' UTR isoform usages on the y-axis, which is similar to the PSI if you are familiar with the alternative splicing. So you can see the difference between usage in two cell types. On the top half, then it means like in dendritic cells, they tend to use more of the short isoform. The short isoform are the major ones. And on the bottom half, those genes, their major isoform are shorter ones in the macrophages. And then when you overlay the differential regulation results on this isoform usage plots, then you can see two interesting things. First, if you look at the genes which are not differentially regulated at all, but still have multiple isoforms, you can see all of them, the difference is very close to 0, suggesting that in order for you to avoid the differential regulation by this microarray, you also tend to avoid any differences in the usages of all the different 3' UTR isoforms. And then on the other hand, if you look at the other genes which are specific to each cell type, then you can see that they're actually spanning on both sides, which if you imagine if all of them are because of the distal site regulation, they actually will tend to be sitting on only one side, not the other, because that's all because of shortening and lengthening, suggesting that that's not the only mechanism. So we are going to look at the second mechanism, the differential regulation of the proximal sites. Here we are doing a similar differential analysis. Instead of looking at two cell types, we are looking in one cell type, but instead we are comparing between both two isoforms of the same target gene, which both contain the same target size but have different lengths. So in this cell type, if you look at the regulation for short and long isoforms, then you can see there's a very similar situation in that sometimes only the short isoforms of this contain the same target size will be regulated, and sometimes vice versa. We're suggesting that maybe the relative positions actually matter. But then is that consistent between different cell types? So if you do the same analysis in B cell, and then I'm using the same color scale, and then map them back to the scatter plot, so you can see that's actually not true. If you look, so in the other plot, you can see that the distributions of those genes with different specificity actually completely change. What used to be specific and now are sitting very close to the diagonal, suggesting they are no longer differentially regulated. So basically, this means like there are some other cell type specific factors that's involved in this process, or to say we're still looking at a mixture of ApA-dependent and independent mechanisms here. So all we have been showing about expression level analysis. So here I just want to show you something very recent result from our collaborators. In this case, instead of perturbing the global regulation by knocking out 155, they're actually knocking in one single 3' UTR for the SOX1 gene, which is a well-known target for miR-155 in some cell types. So in this case, only the interaction between 155 and SOX1, one single interactions will be changed. And then they look at what the phenotypes, how the different functions in different cell types are affected. Then you can see that actually this one single regulation with a more precise preservation are still quite diverse across different cell types. Sometimes it's really effective, sometimes it doesn't change at all. Also suggesting on functional level, those kind of differential regulation also matters. So yeah, that's it. I hope with this talk, I already convinced you that instead of saying gene A is the target for gene B, we should be more careful because that's actually only true in some particular cellular context. And all those mechanisms we discussed today, they're all relevant, although we haven't successfully quantified and decomposed them, the contribution to all the specificity. And also we also want to look at the more detailed mechanisms, like how the RNA binding proteins, different cell types actually involved. And here I'd like to thank both the Leslie Lab and Distal Lab who helped me both on the computational and experimental sides. And here's the funding agencies. I'd like to take questions if you have any of them.
We have time for one question. Ella?
There's someone younger than me. They get it. No, this is a lovely story because it's been known for a long time that these transcripts would polyadenylate at an earlier site when the cells were activated. But in the case where you seem to have the result coming out backwards in one cell type, is it possible that the miR-155 is actually controlling the expression of another miR involved in a network and controlling another miR, so that in fact, when you remove 155, you're now increasing the level of a different miR that can then target? Because do you have any insight into why should there ever be an increased degradation of one of the RNAs when you take away a miR?
Yeah, so I think you're basically asking if there's possibly secondary effects of the miR-155, is not because of direct regulation, but instead because of secondary or more layers of regulation. Yeah, I mean it's possible to check. I think if we actually add in some other data sets, like the CLIP, for example, the CLIP-seq data, which directly measures the binding of 155 to a target gene, instead of right now just using sequence, then we can actually tell if it's directly binding to genes which is regulated, or if it's not binding, then it's more likely to be a secondary effects. We're actually working on those kind of data with our collaborator, but yeah, they're not here yet. I mean, if you got those kind of data, then I guess we can better discriminate between the first order and then second, third layer of effects.
I think this is very important, though, because it really affects the way you interpret this. And one person has mentioned to me a different mechanism where the miR could actually protect against one of these proteins that are involved in rapid degradation of the UUA sequences. And so that might be an interesting thing to look for in some of those cells.
Sure. Thanks a lot.
Wonderful. Okay. Our next speaker is Tom.